Welcome to ISL-enabled Virtual Lab of CSIR.
Swasti Lab's lab in CCMB works on structural and functional integrity of proteome and related protein aggregation diseases.
Let's talk about the central dogma of molecular biology. Genes, these encodes the information as well as stores the information. mRNA that transcribe the information for the proteins to execute the function. In order to function properly, all protein needs to acquire a particular conformation. We call them protein structures. Sometimes for multiple different kind of reasons, proteins cannot achieve their functional structures, and then we call these proteins are misfolding and forming aggregates. These kind of aggregates are associated with multiple different age-related degenerative diseases, including Parkinson's disease, Alzheimer's disease, and so on.
One such protein aggregation disease is Parkinson's disease, where a protein called alpha-synuclein forms protein aggregates inside the neurons of patient brains. So this neuron degenerates and therefore causes the disease. So here we are trying to understand how these aggregates affect or causes this protein homeostasis disturbances. So we are modeling these protein aggregates in the primary neurons, which are isolated from the mouse brains or human derived pluripotent stem cells. What we have shown here is like this aggregate basically interacts with the nuclear envelope and destroys the nuclear lamina, and that is one of the main cause of the disease.
So this nuclear lamina injury makes the neuron susceptible for various kind of stresses. During aging, the cell undergoes multiple stress which increases the toxicity of the aggregates in the neurons and hence leads to neurodegeneration. So understanding the mechanism of this lamina association with these aggregates, devise a strategy so that we can prevent this association and hence decrease the toxicity of these aggregates in the patients.
We also study protein aggregation diseases related to mitochondria. Mitochondria, which is a powerhouse of the cell, harbor respiratory complexes which help in cellular respiration and ATP generation. Respiratory complex 1 is the largest of respiratory complex. It is an ancient protein made up of 44 subunits in human, while only 14 subunits in bacteria. Different organisms which survive in different environmental conditions rely on complex 1 for respiration. Recently, our lab became interested in studying how complexity and structure of complex 1 have evolved in course of evolution to function optimally in different environmental conditions.
So the proteins that make up these respiratory chain complexes are encoded by both nuclear and mitochondrial genomes. A balanced protein synthesis, mitochondrial import and degradation of these proteins are important for the biogenesis and organization of these complexes. Our broader goal is to understand how each of these processes contribute to the complex assembly.
Our lab has also shown that the individual subunits of complex 1, they are aggregation prone inside the cell. We have also shown that these aggregation prone regions of these proteins assist the individual respiratory complexes to associate with each other to form supercomplexes that improves the respiratory function. We use various cell biology, microscopy and proteomics tools to understand the complex organization of these proteins, because mutations of these proteins destabilize this organization that leads to various protein folding diseases.